Today we're going to start chapter 13, where we take the idea of Mendelian genetics and we bring it forward into the 20th and 21st century. Now, as I stated on the chapter page in Canvas, I really don't like the way this chapter is written in the textbook. It has a lot of this, the information there. It's not laid out in a way I like, and they skip over some things that I think are a little important, and focus on things that I think are less important. What can you say? You get what you pay for. It's a free open source textbook. What that means is that instead of one author who wrote everything, what they have is different professors from all over the country, maybe even the world, who will write different parts and submit to be added to the chapters. And, well, professors are going to write about the parts that interest them. Maybe it's their research, maybe it's their specialty, what they specialize in. So they'll write this part, and that's what gets focused on in that section of the chapter. Whether it's something that's a major key point in other textbooks or not, well, that's what's been submitted, so that's what they'll go with. So what I have done is I have taken the lecture slides from a previous semester using a different textbook, and that's what I'm going to use here in the lectures. So do keep this in mind. It's the same information. The information is still presented in the chapter. It's just here it's going to be presented in a different way, a way that I think is a little bit better. I like this textbook the way they've set stuff up. I know some of you are asking, well, if this textbook is better, why aren't we using this one? The one we're using now, free. The textbook that we had previously used, a couple $100. So, not so great a textbook, but it's free. Really good textbook, but oh my God, it's like a car payment or two. Okay, so we're going to go with the slides from a different textbook, like I said, different semester, and I will point out the stuff that's appropriate so that you see that it is still in the same chapter. When we are talking chromosomal theory, the theory of inheritance, the chromosomal theory of inheritance, however the textbooks phrase it, we have to keep in mind that this is nothing more than an extension on what Mendel found in his pea plants, the numbers, the patterns. We have to fast forward a few decades after Mendel published his work, and we see that there are other scientists, okay? They are scientists, okay? This is now in the realm of the scientific method. Other scientists are beginning to look at how things are passed on in other organisms. They are taking a scientific approach to this, the study of what does it take to get the characters, the traits passed on generation to generation to generation. Two of the early ones that we're going to focus on this slide, Bovary. He studied sea urchins. You can read a little bit more about this in the textbook and do the little Google. Do the Google. You'll see that Bovary studied sea urchins, and he studied how these things we now call chromosomes. They referred to them as chromosomes. Didn't know what they were. Still didn't understand DNA. Still didn't understand how genes are encoded within the DNA strand. All that stuff. That's still 4 or 5 decades to go. But what he was able to show, using microscopes and using, different techniques, he was able to show that these things that were called chromosomes that you could see in stained slides and stained cells, that unless they were passed on via gametes, the progeny would not develop. The gametes had to have the chromosomes in them. They lacked chromosomes. Gametes just did not fuse, did not form the zygote, would not make the next generation. Sorry about that. Phone started going haywire. People just blowing it up. Woohoo, Dr. Welsh, Dr. Welsh. Oh, of course, my family doesn't call me Dr. Welsh. They call me, hey, dumbass. No, joking. So, Bovary, sea urchins, these chromosome things, and a lot of his work is also based on the work of the second person we marked here, Sutton. Sutton is actually one of the first to study what we now call meiosis, and the lining up of the tetrad, the separation of the homologous, and the separation of the sister chromatids in meiosis too. He was one of the first to actually begin characterizing this. Still don't know what chromosomes were, how they function, just isolate them. There's proteins, there's sugars, and there's some nitrogenous compounds. Basically all I had then. But what we've been able to build on over the next decades is Mendel's work. What we have found is that Mendelian genes, what he called characters that had different versions he could refer to as traits, what we now call genes, and having different alleles. Every gene has a specific address, okay? We call it loci. Why do we call it loci and not address, like my analogy? Well, because it's going to be a series of numbers, okay? It's not going to be an address. 1312 Main Street. Okay, 1312 Main Street, you know where that is. Okay, you can look on a map. The maps for loci is going to be a little bit different. Okay, because there's no discrete gene 1, gene 2, gene 3, person to person. Yes, there's always going to be gene 1, 2, 3, but there may be some intervening stuff that's going to be slightly different depending on whether you're Northern European heritage, African heritage, South Pacific heritage. So we come up with this idea of loci. Loci are going to give us a rough estimation, somewhere, on chromosome 7, between around this nucleotide region. It should be the gene for blank. Based on the work of Sutton, we've also begun to show that Mendel was correct. Where he was discussing in his characters and traits, independent assortment, segregation, we were able to show, and we discussed in chapter 11, individual chromatids line up, the homologous separate, the sister chromatids then separate. So over the past century, we have studied, then we have identified the different proteins that hold the tetrads together. We have identified the motor proteins and the kinetochore that allow for anaphase 1, anaphase 2 to occur. So we've been able to prove Mendel was right. Like I said last chapter, Mendel, crazy, stupid, smart. So when you go and you look at Mendel's work, you can see that what he talked about, his yellow round, his green wrinkled,
big Y, big Y, big R, crossed with homozygous little y, little y, little r, little r. The F1s, as we see here, all look like the homozygous dominant, the double dominant, yellow round. But we see that it's double heterozygous. But how things line up. Reds to one side, blues to the other, or a mix, will give us the different gametes that will lead to a 9331 F2 offspring.